Good morning. So today we are going to discuss about the anti-hyperlipidemic drugs. Now, before we go to the anti-hyperlipidemic drug, first we need to understand about the different plasma lipoprotein lipid biosynthesis process. Now, lipids are carried into the plasma in lipoproteins after getting associated with the several apoproteins. So here, lipoproteins are bind to this apoprotein, and then you can say that they are carried to the plasma. Now, plasma lipid concentration are depend on the concentration of the lipoprotein, because again it is in the first line we have told that they are going to associate with the lipoproteins. The core lipoproteins globules consist of triglyceride, all cholesterol ester, while outer polar has phospholipid, free cholesterol and apoprotein. So here we can understand that the core lipoprotein globules, clear? Core lipoprotein globules, it is consist of the triglyceride or that is cholesterol ester, and the outer part, that is the polar in nature, has the phospholipid, free cholesterol and apoproteins. Now if we are seeing about this chart, these are the different classes of the lipoproteins. Clear? So first one is the chylomicron, which diameter is 100 to 500, and lipid. See here the triglyceride is major part is the triglyceride, and we can find that is cholesterol ester is less, and main source of the lipid diet and function is for the triglyceride transport. Next one is the chylomicron remnant, and this is 30 to 50. Again here we can find that cholesterol ester is the main thing compared to the triglyceride. Again the diet and the chylomicrons is the source, and dietary cholesterol transport. Next is VLDL. Now, in case of the VLDL, we can find that the diameter is the 40 to 80 nanometer, and here again the triglyceride is the major and cholesterol ester is the minor, and it is major source is the lipid, and it is function is the endogenous triglyceride transport. So we have seen this very low density lipoprotein. Next one is the that is ideal intermediate density lipoprotein. Again 30 to 35 is diameter in nm, and here we can find that cholesterol ester and triglyceride is there. The cholesterol ester almost similar or a little bit more than the triglyceride, and source of this lipid is the VLDL, transport of the cholesterol ester, then triglyceride to the liver, and the source of the LDL. Next is the low density lipoprotein or the LDL, which is 20 to 25 nanometer in diameter. Lipid contains, that is the again cholesterol ester, here no triglyceride, and ideal is the source, transport of the cholesterol to the tissue and the liver. Next is HDL, high density lipoprotein. Now that is the 5 to 10 diameter in nm, and phospholipid, and the that is the cholesterol ester is the in the you can say that lipid content, and source is the tissue and cell membrane, and function is the removal of the cholesterol from the tissue. Now here we need to understand that whenever we are talking about the HDL or the high density lipoprotein, it is considered as a good cholesterol. It is considered as a good, whether in case of the LDL, VLDL, all those things, it is considered as a bad cholesterol. Now next is we will see about the metabolism of the lipid. Now in case of the metabolism of lipid, what we can find if we see about that. Now liver is the central part of the processing side, central part, or we can say that major processing part for the lipoprotein metabolism. Lipoprotein metabolism. Now cholesterol, whenever we are talking about the cholesterol mainly derived into the liver through main three sources. Three sources. So what are the three sources? First one is the biosynthesis of the cholesterol from the acetylcholine. Next is the dietary, you can say that cholesterol by chylomicron RAM10, and next one is the that is the endocytosis of the low density lipoprotein cholesterol by LDL receptor. LDL receptor. So that we have seen that there is a major three process or source of the cholesterol in the liver. Now, triglyceride formed in the liver. Clear? Triglyceride formed into the liver from fatty acid. Clear? Fatty acid, which are derived from the you can say that lipolysis of that triglyceride, or you can say that lipolysis of the triglyceride in the adipose tissue. Clear? In the adipose tissue. Clear? So this is the adipose tissue, lipolysis of the triglyceride. So it will cause the fatty acid which is entering into the blood, and then you can say that this fatty acid entering into the liver, and then it will be transforming to the triglyceride. Clear? Triglyceride. Now triglyceride, cholesterol and protein. Clear? Triglyceride, cholesterol and protein, it is packed together in the Golgi you can bodies in the liver to form the very low density lipoprotein. So this triglyceride, protein, cholesterols, it is producing the very low density lipoprotein. And like you can say that here this is the all those things, apoproteins are there, like cholesterol, cholesterol ester, all those things also it is there, which is denoted here. And then this VLDL, it is secreted into the circulation. Clear? It is secreted into the circulation. Next is this VLDL accept apoprotein CE. Clear? Apoprotein CE, it is accept from the high density lipoprotein HDL. Clear? High density lipoprotein HDL, it will accept the you can say that apoproteins, which is C and apoprotein E, and then return this apoprotein to the HDL as they delivered you can say that triglyceride to adipose and the other tissue via action of the lipoprotein lipase. Clear? Via action of the lipoprotein lipase. So that we have we can see here. Clear? That we can see here, and this is the lipoprotein lysis. So VLDL again it goes into the adipose tissue as a triglyceride. Clear? With the removal of these apoproteins and triglyceride, VLDL are transformed into LDL. Clear? Now VLDL, it is transforming into LDL, and you can say that through what? Through intermediate density lipoprotein. So here ideal and then LDL. Clear? Then first one is the ideal and then LDL. Now you can say that this LDL deliver cholesterol to the various site of the body. Clear? Various. Why we are talking about the various site of the body? You can find it here, the peripheral tissue also. LDL is delivering the cholesterol. Again we have told about all the things into the liver, including the peripheral tissue as we have already told, and also in the liver via endocytosis in the LDL receptor. So LDL cholesterol is also incorporated into the atherosclerotic plaque. Now, if we see about this atherosclerotic plaque, what we can observe this? See, in case of the atherosclerotic plaque, whenever we have discussed this in case of the last classes also, that is like in case of the angina pectoris, whenever we have told about this atherosclerotic plaque. So this is LDL, it is important. Now this LDL, whenever we are talking about this LDL transport to the cholesterol. Clear? LDL transport to the cholesterol to the peripheral tissue for the incorporation into the cell membrane and the steroid. Now in this process, when this particular process it is going on, in this process LDL bind to the specific LDL receptor. See here you can find it is bind to the specific LDL receptor that are located into the plasma membrane of the cell and recognize the apoprotein B100. Clear? Apoprotein B100 on the surface of the LDL molecule. After binding, clear? After binding, we can see that after you can say that they will incorporate after binding this receptor LDL, it will undergo the endocytosis and incorporate into the lysosomes. Clear? It will incorporate into the lysosome for the further processing into the cholesterol and different proteins. Now this LDL can also able to deliver to the nascent atherosclerotic things and thereby contributing into the atherosclerotic plaque. So this is all about the means metabolism of the lipid, that we can understand into the metabolism of lipid. So next we'll about see about the whenever there is a increased level of this triglyceride, cholesterol, etc. You can we can that is we are calling as a hyperlipidemic condition. Clear? Hyperlipidemic condition. Now we'll see that what are the drugs is used for the treatment of the hyperlipidemic situation.